Thank you, everyone, for joining today's webinar. This webinar will be recorded and your phones are muted. So at any time, if you have any questions, feel free to use, the chat box or the Q&A session. You can direct your questions directly to myself. This is Dana Haley Vicente, or the DiscoverX marketing host. The floor will be open about 5 minutes after the webinar is concluded if you have any questions. And then if we do not get to your question, we will respond via email using the email address you provided when you signed up. The emails displayed at the last slide, you can email one of us as well. We will send the recording, the visual recording, as well as the slides after the webinar. So let's get started. Today's webinar is called Application of Beta-arrestin Assays to the Orphan GPCR World. And our presenters today are Arturo Oshii and Ralph Jockers. They are both from the University of Paris-Descartes in CERN. And we would really wanted to thank them for presenting today. They will be talking about how to apply beta-arrestin assays, to characterize orphan GPCRs, and basically how to design your beta-arrestin assay for receptors with spontaneous activity, and go over discussions on heterodimers, allosteric modulations, etc. So with that, I'd really like to thank the two speakers and let them get started.
Thank you very much. Thank you very much. Hello, this is Ralph Jockers, and it's my pleasure to present you some of our most recent results together with my colleague, Dr. Oshii, and particularly on different beta-arrestin assays that we developed and for orphan GPCRs. So when we talk about GPCRs, we are talking about approximately 800 receptors in humans. They can be classified according to different classifications. And the point that I want to make here is in almost all of these subclasses, there are quite a substantial amount of orphan receptors still. So orphan means there's no cognate ligand known for these GPCRs. And if you sum up all of them, you reach a number which is quite substantial, around 100 orphan GPCRs. And it's important to deorphanize them to better understand the function and obviously also to initiate drug discovering programs. So today we would like to concentrate on the class A or rhodopsin-like branch. And as you can see here in this slide, this is the phylogenetic tree of the different members of this rhodopsin-like family. And we would like to focus even more on a subfamily, which is a melatonin receptor subfamily, which is composed of the MTNR1B and A gene, which are actually coding for the MT1 and MT2 melatonin receptor, binding to this hormone, and an orphan member, which is called GPR50. So apart from this genetic and phylogenetic link between these receptors, there exists also a physical link, which means some years ago we could show that these proteins can form heteromers. So this can happen between MT1 and MT2, as you can see here, where the heteromer is actually essential for a physiological function which in the retina, so photoreceptor sensitivity during the night. Moreover, we can also have heteromeric complexes between the MT1 receptor, which is normally binding to the melatonin hormone, and the orphan member, which is GPR50. So in this setting, actually we have an orphan receptor, which is an allosteric modulator of the MT1 receptor, as within this complex, GPR50 abolishes the capacity of MT1 to bind to melatonin or to the G protein or beta-arrestin. This can even go further in the third example, where the melatonin receptor MT2 is heterodimerizing with yet a more distant GPCR, the 5-HT2C receptor, so a serotonin receptor. So here we are in a configuration where we have a transactivation where melatonin can activate the signaling pathway of the serotonin receptor. This is of particular clinical importance, since we think that this heterodimer is involved in the antidepressant effect of a clinically used antidepressant, which is argumentin. So coming back now to our phylogenetic tree, today's focus will be yet 3 further GPCRs, orphan GPCRs, that have been classified recently as potential new members of this subfamily. So we wanted to know whether this is the case, whether they are binding to melatonin, and whether they have any relationship like, for example, heterodimer formation. So the 3 members that we are interested in are GPR 61, 62, and 135. So the results of this work have been recently published in papers published in scientific reports. And this work showed 3 main findings. The first finding is we could show that melatonin is not the cognate ligand of these 3 orphans. However, we could show that these 3 orphans have a substantial ligand-independent signaling activity. And these 3 orphans are actually also forming heteromers, and by doing so, allosteric modulating, particularly the function of MT2. And we will go into this in more detail later. Before doing so, I would like to introduce you the different beta-arrestin recruitment assays for GPCRs. So we have the classical assay that is based on the BRAT, bioluminescence resonance energy transfer technique, the microscopic assay, and thirdly, the enzyme complementation assay, the pass-hunter beta-arrestin assay that is has been developed by Discoverix. So if we go through this slowly, there are advantages and disadvantages as always. So the main advantage maybe of the BRAT assay is that we have access to kinetics, so we can do real-time measurements in living cells, and it's quite quantitative. The obvious advantage of the microscopic assay is that we have access to the compartment, so we can see in which compartment the interaction takes place. And the main advantage of the enzyme complementation assay of Discoverix is its high sensitivity due to the cumulative endpoint measures and also the quantitative character. Disadvantages on the other side can be, for the BRAT and the microscopy assay, the sensitivity can be an issue, doesn't have to be. For sure, the microscopic assay is not really quantitative, only semi-quantitative. For the enzyme and complementation assay, obviously as an endpoint assay, we don't have access to the kinetics. And in some cases, not in all, we might have to optimize the linker for this assay. So let me now a little bit concentrate further on this last assay by introducing you the principle. So you have your GPCR of interest that you fuse to a small portion, which is actually a portion of the beta-galactosidase that is called here in this case prolink tag. On the other side, you have beta-arrestin, which is fused to the remaining part of the beta-galactosidase, which is here called, the enzyme acceptor. So upon recruitment of beta-arrestin to the receptor, you will have also proximity between the prolink tag and the EA, which then complements and then reconstitutes a fully functional enzyme, which then can convert the substrate into the product. And one of the products is luminescence, which can be easily detected by luminometer and very nicely quantified. So with this assay, actually we asked several questions and applied this assay to our specific questions. So the 3 main chapters that will now follow is the first question. So yes, can we screen for the ligand? Can we identify the ligand with the pass-hunter beta-arrestin assay? The second will be rather a study on agonist-independent beta-arrestin recruitment. It's quite a new and different issue that we applied to these orphan GPCRs. And the third issue is related to allosteric modulation of the melatonin-induced recruitment to the MT2 receptor, which can be regulated in heterodimers by these GPCRs. So with this, I will hand over to my colleague, Dr. Oishi, who will guide you through the different experimental parts.
Hello, everybody. At first, we would like to show the ligand screening assay by pass-hunter beta-arrestin assay. because the start of this project is that these three GPRs are classified to melatonin receptor subfamily. So we checked whether melatonin is a ligand or not to these GPRs. As shown here, these three GPRs don't respond to melatonin through path-hunter beta-arrestin 2 nor 1 recruitment assays. We also performed radio-labeled melatonin binding assay and several signaling assays, such as cyclic MP or IP1 pathway, and melatonin does not show any positive results in these assays. So we concluded melatonin is not the cognate ligand for GPR 61, 62, nor GPR 135. Although we were unable to deorphanize our orphan GPCRs, there are several successful examples which demonstrate this assay is useful to deorphanization. These two publications revealed natural ligand of orphan GPCR, GPR 32 and GPR 18, and other groups also found several surrogate ligands of several other orphan GPCRs by path-hunter beta-arrestin assays. So we think this assay will be still useful for ligand screening for orphan GPCR. All we are talking about part 2, agonist-independent arrestin recruitment. The expression of all three GPRs showed beta-arrestin 2 and 1 recruitment by path-hunter beta-arrestin assays. To make sure these results reflect beta-arrestin recruitment on GPCRs, we think control experiments are very important. As a control experiment, we'd like to propose 3 experimental settings. First is negative control by expression of prolink tag PK2 alone. Second is competition experiment with unlabeled beta-arrestin. And the third is confirmation of beta-arrestin recruitment to GPRs with other assays, such as immunofluorescence or BRET. We didn't show the first part today, since the negative control value is very, very small. So here we would like to introduce from the second example. The principle of path-hunter beta-arrestin recruitment assay is association between prolink tag of GPCR and enzyme acceptor of beta-arrestin. So if beta-arrestin without enzyme acceptor exists in these conditions, it's going to work as a competitor of the beta-arrestin enzyme acceptor. As you can see, the expression of beta-arrestin YFP didn't modify the expression level of beta-arrestin enzyme acceptor. This expression of competitor decreased luminescence signal, which suggests interaction between beta-arrestin 2 YFP with GPR PK2. This confirms the luminescence signal due to expression of GPR reflect the association between GPR and beta-arrestin. Other ways to confirm agonist-independent beta-arrestin recruitment on GPRs are other techniques such as BRET and immunofluorescence. Here, instead of BRET, we only show the immunofluorescence, and we can see co-localization between GPRs and beta-arrestin. Furthermore, these GPRs co-localized with internalized transferrin, which suggests GPRs localized in endosome as a result of internalization. We are thinking these several methods are necessary to confirm agonist-independent arrestin recruitment on GPCR. The third part of our findings is allosteric modulation of the melatonin-induced MT2 signaling. Here, we showed the effect of GPRs on ligand-induced beta-arrestin 2 recruitment to MT1 and MT2. Because GPR50, another classical member of melatonin receptor subfamily, is known as a negative regulator of melatonin receptor type 1, MT1 signaling, so we examined whether these 3 GPRs could affect melatonin signaling through MT1 or MT2. The co-expression of GPRs didn't modify melatonin-induced arrestin-2 recruitment to MT1, but co-expression of GPRs inhibited melatonin-induced arrestin-2 recruitment to MT2. In this experiment, receptor expression level is monitored and judged as equal in each condition. Then we examined effect of GPRs on ligand-induced G protein signaling. Since both MT1 and MT2 are GI-coupled receptors, we checked the effect of the co-expression of GPRs on GI cyclic AMP pathway. But these GPRs don't modify neither MT1 nor MT2 GI cyclic AMP signaling. One of the possible explanations of this allosteric behavior would be formation of heteromer between GPRs and MT2. We examined heteromer formation by breath and co-immunoprecipitation. In breath, co-expression of MT2 and GPRs made a nice saturation curve, but with negative control of CCR5 didn't make it. This suggests GPRs and MT2 make heteromer as a specific combination. Co-immunoprecipitation also supports this heteromer formation. So this heteromerized GPRs or MT2 might be responsible to this allosteric modulation. For further mechanism, the knowledge of recent publication from Lefkowitz group helped us. In this paper, they showed GPCR and beta-arrestin interaction consists of two steps. As a first step, C-tail of GPCR is recruited to beta-arrestin. Then, as a second step, finger loop region of beta-arrestin is inserted into GPCR and make much stronger interaction. Coming back to our GPCRs, we hypothesized that these three GPRs inhibit the MT2 C-tail recruitment to beta-arrestin. To examine our hypothesis, we established MT2-MT1 C-tail chimera protein because MT1 arrestin association is not inhibited by GPR. So, if the C-tail of MT2 is important to this inhibition by GPR, this chimera should not be inhibited. But on the contrary to our expectations, even this chimera MT2, its association with the arrestin is inhibited by GPR. So this suggests C-tail of MT2 is not critical for this inhibitory effect. Currently, we are thinking GPR possibly inhibit the second step of the interaction between MT2 and arrestin. This would even make more sense since the second step is supposed to be more sensitive to this allosteric regulation within heterodimer. So, let me summarize our findings. Recently, these three GPRs are proposed to be classified to melatonin receptor subfamily, but they didn't bind to melatonin. However, they made a heteromer with MT2, and they inhibited melatonin-induced beta-arrestin recruitment in the allosteric manner.
So let me now conclude more generally. I think it's of quite interest to see that this beta-arrestin assay can be, first of all, be used for ligand-independent beta-arrestin recruitment and ligand-dependent beta-arrestin recruitment. The ligand-independent assay is quite tricky because you always have to distinguish between the background, the intrinsic background of the assay versus the constitutive recruitment, which is receptor dependent. So we propose to some of the control experiments that might be helpful to distinguish this as this negative control like the ProLink tag alone, the competition assay, or also I think very important completely independent assay like the immunofluorescence, BRED, and other assays. we could furthermore show you that this assay is also useful to deorphanize GPCRs, which was not our case, but has been shown in the literature for a lot of cases. And lastly, this assay is also able to pick up allosteric modulations. So that was our case, where we could show that our orphan GPCRs allosterically regulate the property of our MT2 receptors to recruit beta-arrestin. And so this, interestingly, was only seen in the beta-arrestin assay, but not in the classical G protein GI activation assay. So there seems to be some biased behavior that could be also though picked up with this assay. And with this, I would like to thank you for your attention.
Great, thank you, Dr. Jockers and Oshie. We've got a few questions. All right, one of them was, did you guys test, I guess this is cyclic AMP and IP3 activity type, pathways with, in your controls?
Of course, we tested this possibility by adding melatonin, so the ligands to be tested. But we also tested this actually concerning the constitutive activity. Maybe you can show, yes, it should be this slide. So interestingly, so this constitutive activity was not only seen for the beta-arrestin recruitment assay, these are the data that you already saw, but so we also considered this possibility for IP1 production or cyclic AMP production. And as you can see, we have some constitutive activity also in these assays. And even more interestingly, it seems to be receptor dependent. So we have a differential picture. So it's 62 has actually constitutive activity in all three assays, 61 in 2 of them, not in the IP3 assay, and the 135 only in the beta-arrestin. So there seems to be a differential picture in terms of activating constitutively the different pathways like cyclic AMP, IP3, and beta-arrestin.
Goodness, yeah, it's like you knew they would ask that question. That's perfect. So somebody asked about the cell type or the cell background that you used in your study, and if you did, I guess, multiple ones, how did the results affect related to co-localization and beta-arrestin? So cell types, basically.
Yes. So we actually typically use the standard fibroblast cells. So our study was performed in HEC293 cells and HeLa cells. Sometimes we are also using CHO cells. Typically, these cell types are quite comparable.
Okay. One was related to beta-arrestin 1 versus 2. I believe you had a slide earlier. So they're just asking, do you see differences in other type of GPCR experiments that you may do?
That's for sure an interesting question, and it was an unknown outcome. So in our study, we didn't see a difference. But yes, of course, we would expect differences for example, Class A or Class B GPCRs where we have differential affinities. In our study, it was actually a question raised by the reviewer to see whether we have differences, but we couldn't pick them up, at least for the constitutive activity. So these, at least for the constitutive activity, these orphans seem to behave like Class B, so a similar positive response to beta-arrestin 1 and beta-arrestin 2.
Okay, so similar. Did you try other ligand types like inverse agonists?
No, so we should have, but this was actually not our main objective. So we had a very targeted approach, whether we wanted to know whether it's melatonin or not. Although we also added known antagonists and inverse agonists of melatonin receptors to see whether indeed we could decrease the constitutive activity, so to give some hint on this side, but we were unable to modify this constitutive activity. So which means that the constitutive activity and the potential ligand-induced activity go both in the same direction. There's no melatonin or melatonin ligand binding activity.
Okay. What I'll do is I'll just take 2 more questions. Let's see. This one's a little more general. It's about heterodimer interactions. What other heterodimer interactions, I guess, besides the ones you mentioned, the serotonin and melatonin ones, have you seen in the GPCR world in general?
Ooh, that's a very
that's a tricky one.
That's a very general question, but let me give a general comment on this.
Okay.
This is indeed something that we are interested, like many labs, since the year 2000, 2002. And yes, so the surprising result was that many of these GPCRs seem to tend to form heterodimers. So there was an explosion of articles, although the question mark is still relevant, because when it comes to endogenous expression systems, then the convincing examples of heterodimer formation are much smaller. So particularly also when it comes to what is the specific function that this receptor is related to. So in this case, I would say there is maybe 10 to 20 reasonably well-established and admitted heterodimers that exist with a reasonably well-characterized function. But so what is really always difficult is to prove that the heterodimers exist and what is the function in the physiological context. That's the point here. So in our case, we could demonstrate this, but there are a lot of cases where the question is still open.
Okay. Yeah, that one was tricky. One last question was related to have you tried site-directed mutagenesis, like studies to see where ligand binding related to your study?
No, we didn't exactly, because actually this would have only made sense if we would have had a hint for the ligand binding. And actually, an interesting side note is that we were considering to mutate relevant residues in the dry motif, rather related to constitutive activity. So the dry motif is indeed modified in this often GPCRs with constitutive activity. So I think this would be interesting to study in the future, particularly considering that we have a quite unique constitutive activity profile. So it's not that one receptor is either constitutive active or not. Actually, the constitutive activity depends on the pathway. So I think the interesting point here would be in the future to measure, when we modify the dry motif, which of the constitutive activities would be affected or not. It's a very interesting point.
Okay. Yeah, that sounded like from a structural biologist.
Yes.
Okay, well, we want to thank you both again for an excellent talk, and we want to, welcome any other questions via email. We'll get back to you. You can email myself or anybody on this slide that you see. And thank you again, and have a nice day or the rest of your evening.
Thank you very much.
Thank you very much.
Bye-bye.
Thank you. Bye.